Hi everyone, this is the rapid tests lab. So in this lab, we were going to do a demonstration about tests that are called rapid tests because you get the results very fast. You get them almost immediately. So these are clinical lab tests that are done every day in hospitals, and they take advantage of the immune system components, which is basically antibody binds to its antigen always. So a Streptococcus antibody will bind to Streptococcus, a Staphylococcus antibody will bind to Staphylococcus, and we're going to learn about two rapid tests in this lab. And I think this lab is really cool because these tests are really done in hospitals. So just to give some background, rapid tests are immunological assays. Assay just means tests that use specific antigen-antibody reactions to identify an unknown organism. So if an antibody binds an antigen, you know that you have, if the antigen is a pathogen, you know you have that pathogen, and you get results within minutes. These are run in hospital labs, doctors' offices. They're very quick, very easy to run, and anyone can do them. In the lab, you are supposed to see a demonstration of a test that tests for Staphylococcus aureus. So you can imagine if someone has a Staph aureus skin infection. I mean, if you were a doctor and you assume they have it, you could do this rapid test. So we were going to do the Staphylococcus aureus passive agglutination test, or another test we were going to demonstrate is a test that tests for Streptococcus pyogenes, which causes strep throat and causes other things too. And this is a modified ELISA. We talked about ELISA in lecture. So we're going to talk about both of these, the Staphylococcus rapid test and the Streptococcus modified ELISA. Okay, I'm going to start off with the Staphylococcus aureus rapid test. So this is a passive agglutination test. And when we learned about these tests, remember, we talked about these in immunology applications in the lecture. We said that agglutination tests, basically if an antibody binds its antigen, you get agglutination, which means clumping. And in this test, we're specifically taking advantage of this assay to test for the bacterium Staphylococcus aureus. How the test works is it is the passive one. So this one uses latex beads. So you have latex beads, which are these big circles that are coated with Staphylococcus antibodies, and these Staphylococcus antibodies with the beads will bind to Staphylococcus, which is the antigen, if they see it. So if the beads that have the Staphylococcus antibody see Staphylococcus bacteria, they'll bind to it and you'll get clumping. And this is how this Staphylococcus aureus passive agglutination test works. So it uses, again, these latex beads that are coated with Staphylococcus antibodies. And what you do is, it's a very simple test. You're given these kits, and in these kits there's these pieces of paper. So you take the piece of paper and you put a few drops of the latex beads on there. Remember, the latex beads have the Staphylococcus antibody on it, the Staphylococcus aureus antibody. So now you have the beads that have the antibody on there. Then you take a sample from someone. Let's assume maybe someone has a skin infection and you're assuming that it's Staphylococcus aureus. You can take a sample, a swab, and then you mix it with the beads. If you see agglutination or clumping, it means that the antibody is bound to the antigen, so that's a positive result. If you see no clumping and it just looks fluid, it means that the negative result, the person does not have Staphylococcus aureus infection. So here's a picture where we did two samples. Both sides have the beads. You can tell that the beads are blue. And then we added a sample from the person, which is the antigen. In this image here, you can't really, I mean, if you were actually to see it, you don't see any clumping, meaning that the sample did not have any Staphylococcus aureus to bind to the beads with the antibody. In this circle here, these little circles are clumps. So the moment you add a sample from someone, you start seeing clumping because the beads with the antibody are binding to the antigen, which is Staphylococcus aureus. So again, this is a Staphylococcus aureus passive agglutination test, which is a rapid test. And so A positive result results in clumping. And just to talk a little bit about this, so this confirms diagnosis of a Staph aureus infection. And remember, Staphylococcus aureus are coccus shaped. So if you were to look at them under the microscope, they're little circles in clusters. These can cause many different infections. If you do get a Staph aureus infection, they can cause skin infections, they can cause surgical and wound infections, they can spread to internal organs, cause many different infections such as pneumonia. They can affect the lungs. They can cause life-threatening sepsis when they get in the blood. So Staphylococcus aureus should not be in a person's blood. And the toxins can lead to food poisoning, toxic shock syndrome, or flesh-eating disease. And again, you can easily do this test in lab. And the positive result of this passive agglutination test is agglutination, which is clumping. The next rapid test we were supposed to do in lab is a test that's a modified direct ELISA, and it's a Streptococcus pyogenes test. So in this test, you were using basically an ELISA on a strip to test if someone has a Streptococcus infection. Maybe you suspect that someone has strep throat. So in your lab, in your hospital setting, you can quickly do this. It's a rapid test, it's a quick test. So how this test works is you have these dipsticks, and also a picture of them in a second. So they're basically little pieces of paper, and on these pieces of paper, there's an area on them that has Streptococcus antibodies or anti-strep antibodies. And anytime these antibodies on the strip see their antigen, which is Streptococcus pyogenes, they bind to it and you get a band on the strip. So with this modified direct ELISA, it's a form of an ELISA. Again, the strips are coated, these dipsticks or strips are coated with Streptococcus antibodies. If you take a sample from someone and it does have Streptococcus, the antibodies will bind the Streptococcus and you'll get a band here. So a positive result on these tests is 2 bands. A negative result is 1 band. If you see no bands here, it means that their test is inaccurate. So it's very similar to a pregnancy test. And so just to give some explanation, the way this test is done is you take a sample from someone. Let's use strep throat as an example, and you add a little bit of like water or liquid in one of these tubes here. It's just a little tube. Then you take the swab sample and you mix it really well to try to get whatever organism or bacteria in there. And then you take this dipstick and you stick it in that sample. So here, one of the bands will always show up regardless, and this band reacts with something in the, I'm not going to say water, in the buffer, just to show that the dipstick is actually working. What if it's expired? So that's why they always have one of the bands that will always show up. Now, the second part of the dipstick here is coated with Streptococcus antibodies, so you'll only see a band if the Streptococcus antibodies bind to Streptococcus. So if the person does have a Streptococcus infection, you'll see that band. And no lines, if you see 0 lines, it means the whole test is inaccurate. Maybe it's expired, maybe you did the test wrong. It could be due to many reasons. That's why we have 1 line that will always show up, indicative of a negative result. 2 lines means that the person does have the infection, so positive result. No lines, you cannot take the test accurately, so you have to redo it or do another test. So again, with this modified ELISA that we're using to test Streptococcus pyogenes, 2 lines indicates a positive result, confirms diagnosis of Streptococcus pyogenes infection, and this causes many different infections. So I use strep throat because that's common, but Streptococcus pyogenes, the bacterium, can cause many, many different infections, similar to Staphylococcus. So they cause respiratory tract diseases, immune-mediated complications. Remember, we talked about glomerulonephritis and rheumatic fever, skin, surgical, and wound infections as well. You can get toxins that can lead to scarlet fever, toxic shock syndrome, and flesh-eating disease. So this bacteria can cause a lot of very bad diseases, but strep throat is pretty common. So the objectives of these rapid tests are to know what rapid tests are. So rapid tests are immunoassays. Assays are tests. Immuno because they take advantage of antibody antigen binding to look for like an unknown organism or unknown agent. Given materials, identify the Staph aureus agglutination test and determine if the test results are positive or negative. With the Staphylococcus passive agglutination test, you use these little blue beads that are coated with anti-Staphylococcus antibodies. A positive result, we see clumping, like in this picture. Negative result, no clumping. For the Streptococcus pyogenes test, we're using these dipsticks or strips of paper. Positive result, we see 2 bands. Negative result, we see 1 band. No bands, the whole test is inaccurate. It doesn't mean negative, it means the test is not working. And then the way that these tests work in terms of antigen antibody interactions, know that for the passive agglutination test to test for Staphylococcus aureus, the antibodies for Staphylococcus are bound to latex beads. For the ELISA, the direct ELISA that we're using to test for Streptococcus, the antibodies are on a certain section of the dipstick. So that's how they work. And then the antibodies, when they bind their antigen for the passive agglutination test, you get agglutination, which is clumping. For the direct ELISA test, you get 2 bands indicative of a Streptococcus infection. And again, if I know some of you guys do work in clinical settings, and I have had my students tell me that this specifically with the Streptococcus test, this one is commonly used in hospital settings. So I hope you guys understand this, and if you have any questions about it because you're not really doing it, feel free to let me know.